Lectures will be posted after class, right? Because they have the answers to the homework. I'm not going to give you the answers to the homework. Remember, you just have to do 80%. You have to try, right? I'm not collecting it. I'm not grading it. It's an all or nothing grade. 5 points if you did 80% or more. No points if you don't do it. There's more than 10 homeworks. Only your top 10 will count, right? For a total of 50 points. So today we're going to do 2 techniques. We're going to do common aseptic transfers that you'll do throughout the semester, right? You'll be moving microorganisms from one place to another. We want to do this aseptically. Anyone know what that means? Without contamination, right? We don't want to contaminate us, our cultures, or the environment. And so you're going to learn that. Hopefully you watch my videos, which will give you a leg up on what you'll be doing today. Each person is going to do, as you see, you each all have your own rack. Each person is going to do all 3 transfers and do a streak plate. Guess what your lab partner is going to be doing while you're doing your work? They're going to be watching you. If I see 2 people working at the same time, I will take points off your next quiz. I'm that serious about it. Once that person's done, then they're going to watch you do yours. Why are we doing this? I can't watch everybody. Your lab partner is there to help you remember to do the steps. So when I demo today, guess who's my lab partner? All of you. So just like I screwed up in the video, I'm going to purposely screw up today. And I want you to tell me that I messed up. And try and stop me before it's too late. I want you to do the same thing for your lab partner. You don't tell them what to do. You watch, right? And remind them. Say, hey, wait, you forgot something. Don't even tell them what they forgot. See if they can figure it out before you tell them. Does that make sense? Okay. We're learning, so it's no big deal if your lab partner has got to remind you a step. This is brand new, completely foreign to you guys, right? Who's done this before? None of you. So I don't expect you to be perfect today. What I expect you to do is help each other and work together, because that's how the real world works, right? Nobody does anything in isolation. If you think you can do everything on your own, you're delirious. Everybody has been helped or helped at some point in their life. Okay? So make sure you're okay with the person you're paired up with. Because otherwise, change real quick before we do that. Okay. So terms, I'm not going to read through all of these. One of them was on your quiz, though, was the bonus. Which one? Pure cultures, right? So we're going to learn a technique today, one of the isolation techniques called streak plate, to try and achieve that. So pure cultures means you have a single species. So you guys have pure cultures on your tables right now. They're in the racks labeled stock cultures. So you'll notice that there are microorganisms growing in those tubes. We use 3 different types of growth media. We have broth, which is a liquid. So of course we want to be careful with that so we don't spill it, right, like I demonstrated the first day. Then we have slants, which are also in tubes. Remember, these are glass tubes, so we want to grab them by the tube, not the cap, because the caps are not secure. So this is agar and food, and it's put on a special tray at a slant to create a slanted surface for us to grow the organisms in. This is going to be the most common type of media that we use throughout the semester, because it's the easiest to store large numbers of cultures very easily. And then we have plates, which we use just not as often as slants. And again, we've got a few broths that we use, but primarily we're going to be using slants. So if it has no growth, which your tubes that you picked up from the front table and your plate should not have any growth, make sure you check them, right, that you don't see any growth, that they are sterile. We're going to do remember when we had done the ubiquity of microorganisms, like little colored or white things, like any abnormalities on your plates. So I already put away one of the other ones. If you're not sure, I'll look at your media for you. But that's the first thing you should always do when you get fresh media, is make sure there isn't growth on it. So Michael had already handed in a plate to me, so right, these are supposed to be sterile, right? It's got growth on it. It was an old plate. I was trying to use up the old plates, but it grew. Okay, so, you know, we do different transfers where we'll move the organisms from a broth to, say, another broth, or from a slant to a slant, or maybe from a plate to a broth, which is one of the transfers we're going to do today. When we introduce organisms into sterile media from another source is referred to as inoculations. The inoculum is the actual material, right? The bacteria that we're picking up. Sometimes you'll pick up a large amount, or sometimes you'll pick up a little bit and you won't really see. A light inoculum most of the time is beneficial. You only need a tiny bit of these. There's microscopic, y'all. You only need a tiny bit to produce enough organisms for your next culture. So you can see that picture I have up on the cabinet there. That's the number of organisms at the tip of a needle, right? And as you guys will experience next week when we start looking at them under the microscope, a little bit goes a long way. If you pick up too much, then you end up with the loop over here, right? Come look at it when you pick up your loops. It is coated in dead, nasty ashes from poor bacteria because students picked up way too much bacteria. And so now this loop is not usable. If you want to bring it to where you can. You guys can pass it around if you want. If your loop looks like that, then you need help. You're picking up enormous amounts of bacteria. I'm going to teach you the right way. So we already said aseptic transfers means that we're trying to do sterile technique. It's not sterile in truth, right? Because we're working with organisms, right? But again, we want to work with just that one organism or just those organisms and not contaminants. Because we already know the air, right? Our breathing can contaminate our cultures. The desktop, our fingers. So we're going to do the best we can to minimize any contamination. So the type of food that we give our bacteria, the two most common basic types that we use is nutrient agar, which contains beef extract, so you know they like to eat what we like to eat. Peptone, which is a digestive protein. Agar is the solidifying agent, right? That's why it's kind of solid instead of a liquid. Trypticase soy agar is another one that we sometimes use. And so these have partially digested proteins, tryptone and soytone. Soy, right? Beans, partly digested. Why would we want to do that? Why would we want to partially break down proteins for the bacteria?
So that's easier for them to digest.
It's easier for them to digest. Makes them grow faster. Exactly. We want them to eat fast and grow fast. So just like, why do we buy ground up baby food? Like if you give a baby a carrot, it would take him like all week probably to eat that carrot. Because he has no what? Teeth. The poor thing is going to be sucking on and gumming it all week long. Right? Wouldn't get any nutrition out of it. So what do? We blend it up. We puree it. Right? So that they can easily take it in. So these are rich medias, right, for just growing, propagating the organisms that we primarily use. We're going to use more complex medias that have different components in them and are going to do different things to our organisms after midterm. We've already looked at one of those types of plates, though. We looked at the blood agar plate, remember, that someone did their mouth swab from, and how the organisms changed that plate. So sometimes the media will be selective, and that one was selective and could show us different growth patterns. So why do we move them from one tube to another? Why can't we just leave them in one tube? Well, that tube, y'all, is like a fallout shelter. with a whole bunch of MREs and the toilet just broke. Would you want to stay there? Eventually you're going to run out of food and the wastes are going to become unbearable. And that's what happens to these microorganisms. As we leave them there for long periods of time, the waste build up, become toxic, could kill them, and they run out of food. So if we want to keep them growing, we want to keep them happy, we have to keep moving them into a new fallout shelter. So typically we use a loop, right, that you guys passed around, or the tip of a serological pipette if you're working with frozen stocks. And I'll show you guys what that is when I start demonstrating. Selective or differential tests identify organisms by their properties. So like I said, that blood agar media was actually selective for gram-positives and allowed us to see if it could lyse red blood cells. And that's why we saw the green discolorization to that plate. It's not on the slide. What are you looking for? Okay. Usually we'll use a loop, but some of the tests we'll use a needle. So notice in the beakers up front, the one we most commonly use, which are loops, are labeled, put them back in this beaker. Needles, which the label, oh, there we go, it's kind of rubbing off, I need a new one. Notice are pointy and go back here. You won't use these until after midterm. Or, if you have trouble with picking up too much organism, I might suggest a needle to help you with that problem. So differential help us differentiate different things like fermentation of sugars by acid production, or the blood agar, the lysing of the red blood cells. Selective, select for a particular group. So usually there's something toxic in that media that will stop the group that we don't want from growing. In the case of those blood agar plates, they contain 2 antibiotics that kill gram-negatives. We use another one that has a high salt concentration and therefore selects for ones that can grow in that environment. Remember, we want to keep the environment out of our samples. That's why we use aseptic technique. That's why we're going to practice and get better and better as we go throughout the semester. We want to keep those pure cultures pure. We want just one organism in there, one species, that's it. Just the one we're working with. Because you can't study stuff about it if it's a mixture, right? How do you know which one's doing what? You don't. They're microscopic, right? We got to keep them separate. Make sure the media does not become contaminated with more than one organism. And we want to keep ourselves safe, right? We want to keep the sample out of the environment. So make sure our possessions in your body doesn't encounter any unwanted organisms. That's why we wear lab coats. That's why we wear our safety glasses and our gloves. Right? Why we put all our personal items in the closet. And our cell phones in our little bags. And then you may be asking, why did you pick up 2 sheet protectors? Well, are you going to probably have some instructions you might want to follow? What happens if you spill bacteria on those instructions then? I got to throw them in the autoclave and throw them away. You might want them. So your instructions for today, you want to put in those sheet protectors. Now the handout you picked up, don't put it in there because you're going to write on that until after you write on it. Anything you're not going to write on or you don't want anything getting spilt on, you put it in your cubby, right? Yep, and you can separate those 2 pages, right, that you had for homework. Although I think you really only need one of them. The one that has the instructions and the drawings, yep. Those are your 2 sheet protectors for use throughout the semester. You're going to put them in your bag with your goggles and your lab coat, as well as your pen. The back side you're going to write on, and I actually gave you a new one today. So leave it out for now. Once you write on it, then you'll put it away. So I have some dry erase markers that I meant to bring in from my office if you wanted to use that checklist, and then you could wipe it off your sheet protectors. So if anybody wants to use those, I might go grab them before you get started. Okay. One of the cultures that you guys have in the stock culture rack is labeled mixed. I actually mixed 2 different organisms into that tube today. Had I not labeled it mixed, you probably would have no idea that that was a mixture. All right, so the white racks that have the plate on top are your stock cultures. Those contain microorganisms. So they have growth on them. And hopefully our streak plates will look something like this. My streak plate doesn't look so good, that one that's on the top and labeled E. coli, because I did it late yesterday afternoon. But I think I do have some isolated colonies. I just don't have the 4 sections, right? So you'll see where we first put the bacteria, and then we spread it out, and we try to get individual isolated colonies. So look at this, is definitely more than one organism probably, right? There's like 3 different colors here. There's red, there's white, there's yellow. That one's a pure culture that you guys are starting with. I'm giving you guys a mix today in a broth of 2, and we've got to try and separate them out. So once we incubate and we come back, it maybe looks something like this. So a single streak, if the inoculum is light, for instance, remember last week from our desktop, right? Because there wasn't a lot of bacteria, we could just rub just that whole thing on that plate and we didn't get an enormous amount of growth. That's not sufficient for the number of organisms you guys have in that mixture tube. We've got to do it stepwise to dilute out the numbers that are being spread onto each section of the plate. That's an isolation technique. This one's called streak plate, and this is the one that you guys are going to practice today. Specifically, we're going to use the streak plate that has 4 sections known as the quadrant streak, which is what you see demoed in both of these pictures, and what I actually performed to make that plate of E. coli that you have. So we're going to draw something similar to this, and this drawing is also on your handout for today. A lot of times beginning students, you'll notice I didn't draw any lines on that plate. I've been doing this for a long time. Beginning students like to have a guide. It's good. When I first learned, I drew on the bottom of my plates too. Right? This grid seems to work the best. I don't quite angle mine like this picture. I make mine more like goal posts, which is appropriate. You know, it is football season. But we're going to have 4 different sections, right? 4 different quadrants to our plate. So we're going to draw on the plate. I'm going to show you how to do that so that you can see it as we draw on the bottom. So that when you look at it, you see something like this. And usually I just write 1, 2, 3. We know that's the 4th one. You're going to take your inoculating loop and you're going to take a sample and you're going to spread, and I call this invisible coloring, right? Because you won't see the organism going on the plate. They're microscopic. You're going to go back and forth and try and cover as much of this first quadrant as you can with your loop. Then you're going to flame your loop, right? That removes all organisms from that loop. Do you go back into the mixed culture? No. You take your loop and you're going to pick up from the surface of this plate from quadrant 1 and move it into quadrant 2. And usually what I do is I start right here at the top and I go back and forth, right, over this entire section. Then you do what to your loop? You flame it. And then you do the same thing from the 2nd to the 3rd, and then for the 4th. I sometimes do a little bit different method here. I'll draw several lines across. Or you can go back and forth into here a couple of times and then just streak down the plate. Can you do either one? But what does a good scientist do? You document what you do. So you can choose, right? But you got to write down which one you chose.
So like where the, like for example, where the black and red touch and the red and blue, you're just getting really like the corner of it. Gotcha.
Yeah. You just want to try and pick up some organisms from this section and bring them to the other section. And I find, again, for beginning students, it's best that you guys, because you guys don't usually get great coverage, is that you just go back and forth from the top all the way to this line. Right? So you start at the top, go back and forth, back and forth, back and forth, back and forth. And that's what I'll demo today. Same thing over here. You'll start at the top and just zigzag all the way down to that stop line. So depending on the circumstances, you might start out with, say, a swab for the first one, but we're going to use a loop for all of it. So when they swab your throat, like when they think you have strep throat, and they stick it in that tube, they send it off to the lab technician rubs your swab onto quadrant 1 and then uses a loop to do the rest of the quadrants. So I draw mine, like I said, more straight, more like this, more like a goalpost. Some people will leave out some of those crossover lines even, right? Again, whatever's going to work for you is what you do. And then, like I said, I usually do the one straight across. So draw, whatever you choose to do, draw it out on your sheet so you know what you did. Right? With practice, like I said, you won't even draw guidelines on the bottom of your plate. You'll be able to keep track of where you are and where you're going. So you can actually get, using toothpicks, 8 sections onto a plate. Like, so if you're using like several different isolates, when scientists, they'll actually divide a plate into 8 sections. And their streaks will literally be streaked lines instead of quadrants. So they'll do one streak with one side of the toothpick, a cross streak with the other side of the toothpick, get a new toothpick, do another cross, and then zig down that quadrant. Then pick up 2 new toothpicks and do it over and over again to fill all 8 of those sections on that plate. So one of the first things that you guys are going to do before you get started today, as I said, you already have some cultures in front of you. I don't want you to pick them up just yet, per se, because you don't have gloves on. Although you could, they're non-pathogenic. Remember to grab the tubes by the tubes, not the caps. So on the slants, take a note of what type of growth do you see. You know, is it kind of see-through? Like this one, sometimes students are like, you didn't give me anything. I'm like, look closer. It's there. It's kind of transparent, right? There's organisms there. Some of them are really white and chalky. I want you guys to start writing down these types of descriptions. Your source, right, for today, right, it says name of organism. That's on the tube, right? The source is it a slant, is it a broth, or is it a plate? Right? The description is things like this. It's transparent, it's white, it's chalky, it's smooth, it's shiny, right? For the broth, where's the growth? Is it all throughout the tube? Is it settled out at the bottom? Is it only at the top? Does it have any color to it? Is it white? So that I didn't realize that list was still here. This is an old list. The last page. And so these are some growth patterns too for the plates. This is our list for today. So you want to jot that down. So number 1 is Staphylococcus epididymis, and that's in a broth. We're going to move it to a slant. Micrococcus luteus is our number 2 culture. It is in a slant, and you'll notice what about it? It's yellow. And then we're going to move that one to a slant. E. coli is on your plate, and we're going to move that into a broth. We're going to put all these cultures at 25, right? But we're going to, as I said, put them in the racks. Let me